Okay, I think we can probably get going. So hi everyone again. My name is Spian Stoichev, and I'm part of the Resin Bio team. Today I have the pleasure of hosting Dr. Ana Martinez del Val from a Novo Nordisk Foundation Center for Protein Research. Before I introduce Ana and her work, just a couple of housekeeping announcements. We are recording this session, and we're going to make it available for everyone to view at their own leisure. So keep an eye out on our social media channels, as well as YouTube and our website for the recording that's going to come up in the next couple of days. Please post your questions in the Q&A section. We'll have plenty of time to address these after the presentation. And also, if you want to have your question live for a little bit of a discussion, this is also okay. So just indicate this with the raise hand icon, and I will activate your mic at the end of the presentation. And so before we start, just a little bit of background about this series and how it came about and the idea behind it. So as an emerging company, we realized quite early on that in order to deliver solutions that are fitting and tailored to the user, we need to work quite closely with the scientific community. And this networking has been really important for us over the years, and that's where essentially the idea behind this webinar series began, where we essentially now want to utilize the networks that we've built over the last few years to provide a platform, particularly for emerging researchers, to come and talk about the work they're doing, the solutions that are generating in the omics space. And hopefully this will promote collaboration across like-minded labs, like-minded researchers, and they can enhance the probability of talking about their research with their peers. And so this platform is open to everyone, not just our current clients and collaborators. And so if you feel like you want to come and chat about your work in future episodes, please drop me an email, and we can certainly involve you in future series. And so for the main part of this, for this particular episode, as I mentioned, I have the pleasure of hosting Dr. Ana Martinez del Val from the Center for Protein Research in Denmark. Ana has broad experience in quantitative proteomics as well as phospho-proteomics, where already during her PhD, she started applying these techniques to characterize stem cells. And at the time, she was based at the proteomics units of the Spanish National Cancer Research Center. Subsequently, from 2019, she has been a postdoctoral research fellow at the Center for Protein Research at the University of Copenhagen. And she's really been instrumental in developing some of the workflows she's going to be describing today in high-throughput spatial-temporal proteomics. So we will hear about the application of chemical fractionation methods coupled to automated sample preparation to profile both the proteome as well as phospho-proteome. And this is all linked to rapid data-independent acquisition-based LC-MS workflows, with the idea to package this into a pipeline that enables really high-throughput analysis and to monitor proteins both spatially, but also across time, and how they're shuttling across different compartments, and using this to regulate signaling in cells and tissues. And so, Ana, first of all, thank you very much for agreeing to come and talk to us today. It's really much appreciated, and I look forward to your presentation.
Can you hear me well? I hope so.
Perfectly.
So thanks for this nice introduction. So as he mentioned, I'm Ana Martinez, and I'm a postdoc in the Novo Nordisk Foundation Center for Protein Research in the University in Copenhagen with the Jesper Olsen group. And today I will present in this seminar a pipeline that we developed within our group that allows us to study in a high throughput manner the spatio-temporal regulation at both protein and phosphoprotein level. So as probably you know, subcellular location is one of the most relevant ways in which proteins regulate their function. Many proteins, in response to certain signals or stimuli, they are modified by post-relational modifications such as phosphorylation that trigger the relocation within different compartments of the cells where they either activate or inhibit their activity or interact with different set of proteins. Therefore, studying spatial regulation is of high relevance, and it's been of intensive research in molecular biology. Although in this field, they mainly rely on techniques such as immunofluorescence that, although highly sensitive and resolutive, they only allow us to study few selected targets simultaneously at the same time. In order to increase the capacity to study spatial regulation in a proteome manner, some groups based on mass spectrometry so that mass spectrometry-based proteomics workflows can allow to obtain highly depth, sensitive, and resolutive maps of the subcellular proteomes. And these three papers that I write here are the most relevant in the field, and they prove that they can achieve subcellular location of almost 8,000 proteins. Although very resolutive and sensitive, these workflows have certain limitations. For instance, they lack the applicability at the signaling layer of the phospho-proteome, or at least they haven't been used at this level yet. And secondly, they require extensive acquisition times and complex experimental workflows that make them difficult to be applied in labs that lack the expertise, and they are also difficult to be used for complex experimental designs that involve multiple replicates, treatment of time points. In order to address these limitations, here we suggest this pipeline in which we combine a simpler subcellular purification approach based on chemical fractionation, coupled to an almost hands-free sample preparation approach based on the use of the Kingfisher robot and magnetic beads, together with fast and sensitive mass spectrometry methods in which we benefit for salt gradients, ion mobility, and data-independent acquisition analysis. Briefly, what we do in this workflow is we start either with cells or with tissue, and we sequentially lyse the cell, releasing different parts of the cellular compartments by using buffer with distinct chemical composition, either with different reagents, such as digitonin, salt concentration, or detergents. So we end up with 6 different fractions, each one containing different compartments from within the cell. And the protein obtained from its fraction is digested using the protein aggregation capture protocol using magnetic beads in the Kingfisher robot. At this step, we take a small aliquot from the samples to analyze the total proteome. And in order to maximize the protein coverage without compromising data acquisition, we use sort gradients coupled to data-independent acquisition analysis and an ion mobility interface, which we recently showed that all coupled together can maximize the number of proteins we can identify in these sort gradients. On the other hand, with the remaining peptides, we perform phospho-enrichment analysis using titanium iMac beads, again in the Kingfisher robot. And we analyze with sort gradients and with a sensitive method. In both cases, samples were acquired, as I mentioned, with data-independent acquisition methods and analyzed in a spectronaut with a library, also known library-free, or also known direct DIA approach. Altogether, this approach allows us to obtain full proteome and phospho-proteome data at subcellular level in just 5 hours of mass spectrometry time. In order to benchmark this approach, we tested this with HeLa samples in quadruplicates. And here in this heat map, you can see mostly the results of all the proteins and phosphosites identified at quantifying our data. One relevant point to arise from these results is that the protocol is highly reproducible. As you can see, the replicates from within the same fraction always cluster together at both levels. And we also, the second main conclusion is that we can find highly enriched protein in each one of these compartments, showing that the fractionation is working. What we last need to know is which kind of compartment we are enriching in each one of these fractions. And to reveal this, we use very well-known protein markers for each one of the most relevant subcellular compartments. And as a result, we see that proteins from the actin cytoskeleton and the cytosol are mostly purified in the first fraction of fraction 1, which from now on I will be calling the cytosolic fraction. Interestingly, in fraction 2, although we still see some cytosolic fraction, we see enrichment of other proteins found in the cytosol, but in complexes, like the ribosome. When we move to fraction 3 and 4, in which we start to use detergents to release the content in the cell, we start to purify proteins found in either the plasma membrane of organelles such as the Golgi apparatus, the mitochondria, the lysosome, and in the endoplasmic reticulum. Although most of these proteins peak in fraction 4, we interestingly find some clusters of protein in fraction 3 for the ER and the lysosome. When we look at these proteins peaking in fraction 3 and compare them for those in fraction 4, we found that in both cases, they belong to the corresponding organelles, but whilst protein from fraction 3 were from within the luminal part of the organelle, proteins from fraction 4 were bound to the membrane. So this is interesting because our protocol then shows that we can differentiate between proteins from within the same organelle, but with different solubilities or distribution. Finally, in fraction 5 and 6, we purify nuclear proteins. Well, proteins from the nucleoplast of the chromatin are mainly purified in fraction 5. In fraction 6, we found more enriching nucleolide proteins. And we see that this profile and highly reproduce at the phospho-proteomic level, so we can interact information from layers. Thanks to that, we can also study a relationship between kinase and substrate in a spatial manner. And we can generate this kind of maps in which we group together kinases by their subcellular location. In this network, we are plotting the kinase group by, as I mentioned, the fraction in which they are purified, which is indicated by the color of the exterior circle. So for instance, these kinases are cytosolic kinases, or these kinases are membrane bound. These are nuclear. And within the circle, we saw through a pie chart all the sites known for this kinase and their subcellular location. In that manner, we can see that some kinases like PRKA1, it's found in the cytosol, but also most of its known phosphorylation sites for other substrates are also cytosolic. On the other hand, we found that EGFR, which is a membrane-bound receptor and a kinase, also phosphorylates in basal conditions protein bound to the membrane. On the other hand, we found other kind of kinase like GSK3 beta that, although this mainly purified in the fraction 2, which is the second cytosolic fraction, we have a substrate found in the fraction 1, or the purely cytosoluble fraction, but also nuclear sites. So once that we saw that with this approach, we can learn about the protein and the phosphosite location within the cell and how to interact within both layers of information, we decided to apply into a real setting and study how protein move in response to a stimuli in a spatio-temporal fashion. For that, we use a model well known in the field, which is EGF stimulation, which in this case, when EGF binds to the EGFR receptor, it's rapidly activated and phosphorylated, which leads to the recruitment of adaptor proteins that otherwise are free in the cytosol. When this happens, the receptor and the adaptor proteins are rapidly internalized into early endosomes for either downstream signaling or for maturation into late endosome for degradation to shut down the signaling cascade. So we decided to use this well-known model to prove whether we can track down this movement in a spatio-temporal manner. To do that, we started with HeLa cells in quadruplicate that we serum starved overnight. Then we treated with EGF and collected samples at 2, 8, 20, and 90 minutes upon treatment. For each one of the samples, we performed the subcellular fractionation protocol, and we measured the total and the phospho-proteome. In total, this experiment comprising 220 and 40 samples, analyzing the mass spectrometer in just 4 days of acquisition time. So the first thing was to look at, in an unbiased manner, which protein were translocating between cellular compartments. And for that, we performed this kind of translocation plots in which the x-axis represent what we call the mobility score. So the higher it is, the more amount of protein is moving between compartments after 2 minutes of EGF. And the y-axis indicate how significant is this translocation between compartments. Therefore, proteins in this quadrant here are proteins that, with highly statistical significance, are moving between cellular compartments. And interestingly, within these proteins, we found the adapters I mentioned before, GRAP2, SIC1, and CBIL, which are a positive control for EGFR activation and movement. When we look at the same events of 8 minutes, 20, and 90, we see that in 8 minutes we can still see these adaptor proteins moving, but they disappear at 20 and 90, which is in agreement with the known transient activation of EGFR. When we look at the profile of these proteins across time, we first see that EGF is mostly purified in the membrane bound fraction as expected because it's a plasma membrane protein. And since the lysosomes are also membrane-based, we cannot separate them. So there's a limitation. We cannot see EGFR translocating, but we can see its translocation as a proxy of these adaptor proteins. So you can see Cbl, SIG1, and GRAB2 at time 0. They are mostly cytosolic proteins, which is the red-orange bar. However, as soon as 2 minutes of EGFR, EGF is activating EGFR, we can see a distribution change completely and the proteins are majoritarily bound to the membrane. We see that this translocation peaks at 8 minutes, but then is rapidly shut down at 20 minutes, and at 90 minutes, the distribution comes back to initial conditions, showing the transient activation of EGFR. Moreover, we can add to this information about the translocation at protein level, information for the phospho-proteomic data. And we were able to map several sites of EGFR that are known to be activated in response to EGF binding. And you can see in here that all these tyrosine sites are rapidly phosphorylated after 2 minutes of EGF, that they remain active after 8 minutes of treatment, but that the phosphorylation disappears at 20 minutes, which perfectly correlates with the translocation that we saw before. Thanks to the depth of our analysis, we can also go further and look for downstream targets of EGFR activation at phosphoprotein level. And for instance, we found that one of these adaptor proteins, SIG1, was phosphorylated in the tyrosine 427. And we found that in the membrane fraction, which is the dark green one, it's rapidly phosphorylated with EGF activation. But interestingly, we found that the cytosolic form, the cytosolic free fraction of this protein is also phosphorylated in the same tyrosine site, which might indicate the existence of additional tyrosine kinase from the cytosol that can activate this protein. Moreover, we can go even further in the downstream signaling pathway, and we found that Jun is rapidly activated in the nuclear part of this protein, showing that the EGFR signaling can go up to the nucleus. So once that we prove that we can use this pipeline to study spatio-temporal regulation in cells, we decided to go one step further and see whether we can apply it from an in vitro system to an in vivo setting. And for that, we use a mice experiment, again in quadruplicate, in which we treated 4 mice with control and 4 mice with EGF for 10 minutes. And then we collected the kidneys and the livers, and we profile the different subcellular fractions, again, at the proteome and phosphoproteome level. We compared the result with the one we obtained with HeLa, and we found that the EGFR protein distribution was the same. It was purified in the membrane fraction. And then, as we saw in HeLa, the phosphorylation is rapidly activated, showing that EGFR is active after EGF stimulation. And we saw in the liver that the phosphorylation level of these sites also increased, showing that the activation is working. But interestingly, we found that this activation is not only observed in the membrane fraction, but also in the cytosolic, in what we call the cytosolic fraction, which is the red bar. We were quite intrigued by this fact, so we decided to explore it further. And if we come back to the model I presented before, I mentioned that when EGFR is phosphorylated, it's rapidly internalized into the early endosomes. Therefore, phosphorylated EGFR must colocalize with markers of the early endosomes. And lucky us, there is very well-known markers of this kind of vesicles, like RAF5A and EEA1. So we look at the distribution of these markers in our data. We found that whilst in HeLa, as expected, these markers for early endosome was purified in the membrane fraction. Interestingly, in liver and kidney, we found that those early endosomes were purified in the first fraction, which validates the phosphorylation we found before. Interestingly, we found that late and recycling endosomes don't follow this trend. So we now are working further in trying to understand why this is happening only in tissues and whether some part of the protocol can be modified to change this. So at this point, we found that our protocol works in vitro and in vivo, but in an already known model. So we decided to use it to learn something new. And in our group, we are very interested in understand cellular response to stress, and the plasticity of cells to recover for the mentioned stress. So we decided to study the response of UOS cells to osmotic shock by treating the cells with 1 hour with 500 millimolar sorbitol. Then we removed the sorbitol and we collected samples after 30 minutes, 3 hours, and 24 hours to evaluate how the cell can recover from this stress. Again, we performed subcellular fractionation and measured the total proteome and the phospho-proteome. In this case, we performed gene set enrichment analysis to study which group of proteins were changing the distribution across fractions in the different time points. Interestingly, when we look at the changes in fraction 2 enriched proteins, we found that the cytosolic large ribosomal subunits were significantly decreased when compared to the control conditions. But most interestingly, when we look at the same proteins in fraction 6, we see the complete opposite trend. So protein seems to decrease from fraction 1, fraction 2, but these ribosomal proteins were increasing in fraction 6. So as probably you know, ribosomes are comprised of two big subunits, the 60S or the large ribosomal subunit and the 40S or the small ribosomal subunit. So since in the gene set enrichment analysis, we only found the large ones, we decided to explore this difference further. And in this plot that you see in the panel B, we are tracking the changes of both subunits across time between fraction 2, which is the ribosome enriched fraction in the cytosol, and fraction 6, which is the nucleoli enriched fraction. So if you look at the yellow lines, the dashed one corresponds to the small subunit and the continuous one corresponds to the large subunit. And in both cases, we see that the percentage of ribosomal proteins in this fraction decrease after 1 hour of osmotic shock, but then it starts to be recovered and reaches the initial condition after 3 hours after release from the stress. Interestingly, when we look at the same proteins in fraction 6, which is the nucleoli fraction, we found that only the large subunits are increasing and then recovering following the same trend as fraction 2, but not the small subunits. So we decided to validate this hypothesis derived from the mass spectrometry proteomics data to prove what the mechanism we think is happening upon osmotic shock. So in this picture, you can see that the way in which ribosomes are mature and released into the cytosol. So the synthesized immature ribosomes are imported into the nucleoli, where they start to collect the ribosomal RNA and maturation factors, and they go through a very complex mechanism until they are released as a mature form in the cytoplasm. And the maturation pathway of the large subunit is separated to the maturation pathway of the small subunits. So our hypothesis is that osmotic shock is somehow altering uniquely the way that this large subunit is mature and exported into the cytoplasm when there is osmotic shock present in the cell, leading to the accumulation of the large subunit within the nucleoli. We decided to validate this further using an independent biological experiment, and we choose to use immunofluorescence, which, as I mentioned before, is the preferred way to study a specific location of proteins. And in this case, we look at the two large ribosomal subunit, RPL10A and RPL22. And in this case, you can see a distribution when cells are untreated, and here which we observe with the fluorescent marker KEMA. They are mainly cytosolic, and we compare with the distribution of a nucleoli marker, in this case fibrillary, that you can see in red. As soon as we treat cells with sorbitol for 3 hours, you can see that the ribosome signal become more condensed. And when we colocalize this condensed signal with the fibrillary, we can see a perfect match. Therefore, the hypothesis that we observed that ribosome seems to accumulate in the nucleoli upon osmotic shock, with information purely derived from mass spectrometry-based proteomics, was confirmed by using immunofluorescence. We decided to explore a bit further to try to understand why the large subunit is accumulating and not the small one, and we perform a northern blot analysis in which we can track down the maturation of the ribosomal RNA. So in this, in the plot from the left, you can see the different maturation precursors of the ribosomal RNA for the small subunit, and on the right, those for the large subunit. And whilst we cannot see any significant difference for the small subunit, we can see a clear difference for the large one, that the line corresponded to the 12S completely disappeared, and we saw a smear of degradation. So our hypothesis is that osmotic shock is somehow altering the maturation of ribosomal RNA, and due to this issue, the 60S subunit cannot be properly mature and exported into the cytosol when the cell is affected by osmotic shock. So with all of this, I would just like to conclude by going over all the conclusions from this talk. So I hope I convinced you that our workflow allows to obtain fast and comprehensive subcellular maps at proteome and phospho-proteome layers, that we applied it to study EGF stimulation in cells and mice, so therefore our workflow can be used to study spatio-temporal dynamics during signal transduction. And finally, we applied it to a complete novel system to study stress signaling response to hypertonicity, finding that hypertonic shock results in a transient accumulation of large ribosomal subunit to the nucleoli, potentially due to aberration in the biosynthesis or assembly machinery. So with this, I would just like to end by acknowledging all the people involved in this project, and also all the people from my group from the constant input and help, and for the funding. And of course, to you all for your attention, and I will be really happy to answer your questions.
Fantastic. Thank you, Anna, for the great presentation. We already have a couple of questions. Maybe I can get the ball rolling with a few from my side. The first one that kind of caught my attention in terms of from a technical perspective is how do you normalize the data across sections? Because presumably you don't have the same amount of protein that's coming across, and the typical total ion normalization is probably not the best way to go there. Do you spike with some known proteins, or
So,
how do you do it?
So that's a good question that we assess in our manuscript that we are preparing. So we consider that we are performing a sequential lysis of the cell. So we are not losing parts of the cell. If we merge all fractions and we correlate the sum intensity of the silk fraction, it correlates almost perfectly with the full proteome. So we consider that, of course, the protein levels will change between fractions, but that doesn't need to be normalized. We only normalize for within the same fraction across time points, but we kept the protein intensities between fractions because that is how they are present in the cell.
Yeah. So to account for the technical variability, just being across the 4 replicates.
So we normalize fraction 1 in time 0 with fraction 1 in time 2, but we don't normalize fraction 1 with fraction 2 because it doesn't make sense to the reality of our data.
Okay. And do you find when you're presenting your workflow in the beginning, do you find differences in how the LCMS was set up between total proteome and phospho-proteome? What is the optimal setup when it comes to gradients and aspect setup for that?
So we apply for this everything that we learned from our MCP paper when we were testing the Exploris 480 and the EvoSep. And at that point, we saw that a gradient of 21 minutes was the best compromise between short time point and coverage of the proteome, especially when using FAMES. For the phospho-proteome, we've done a lot of implementation and optimization in our group. And for the amount that we are working in here, because we are very limited in amount. So when we fractionate the cells into 6 fractions and we do phospho-proteome, we are already doing phospho-proteome with really small amounts. So in this case, our gradient benefits us because we don't dilute the signal. So it was, that was the main 2 reasons.
Okay, awesome. So we have some questions in the chat here. I'm not sure if you can see it on your side. Do you want to?
Yeah, I think I can. So someone anonymous asked whether is it possible to use this protocol to evaluate the lipid draft proteome. I don't see why not. The main explanation behind our protocol is that proteins are separated based on their ionic interaction and solubility. So if proteins from within the lipid raft, with I guess they have very specific solubility and interaction conditions, probably they will elute in one particular fraction. So I will say go ahead and try. And then there is another girl preview maker asking whether our EvoSep. So it says in our bioarchive paper, it was interesting to see that you use 15 centimeter column with the 60 SPD method. The standard okay, so he said that the standard configuration recommended from EvoSep for the 50 centimeters to use the 30 samples per day.
Okay.
So that is for the PepSec column, the commercial columns. It's true that for the 15 centimeters, the best setting is 30 samples per day. But in here, everything was done with homemade pool columns. And in our hands, the 15 centimeter columns works as well for the 60 and for the 30 samples per day. But it's just because it's our specific homemade columns.
So you can juggle between the two methods with one column, essentially.
Yeah.
Okay. I see that I have one raised hand by Ruth. So I'm going to see if I can activate your mic. So Ruth, do you want to ask your question? Wait, hold on 1 second. Can you unmute? You're going to have to unmute your mic. Or maybe that was an accidental hand. Okay, I don't see any more questions. Maybe one from my side again. Cells versus tissues. There's a very difference of the composition and characteristics. Do you find that you need to change the fractionation protocols to when you go from cells to tissues?
So the main challenge is that tissues are within a tissue, like the cells are not single cell, they are interacting with matrix, and so we need to release that interactions for this workflow to work. And so what we did was just we included an homogenization step to break down the tissue, and we used the Precellis system with an hypotonic buffer. That is something that is being used in many other publications for homogenizing cells from tissues. And then we apply the workflow, the fractionation workflow as it is. So we think that probably because of that, there is some difference in the profiles of cell from cells to tissues, like the example I saw with EGFR phosphorylation and early endosomes, which is not necessarily a bad thing, it's just different. But yeah, mainly is that different, the homogenization step, that if someone really wants to keep the cell intact, maybe they can use some of these workflows used for single cell genomics or proteomics, where you can like break down the interaction without breaking the cell. But we never tested that.
Okay. There's a question in the chat from Luke. I don't know if you can see it. Do you standardize the amount of sample of each fraction for phosphopeptide enrichment?
So the same as I mentioned before, we want to keep knowing that the distribution of intensity is across fractions, so we cannot standardize, otherwise we will lose it. We do the phospho enrichment with all the peptide recovered per fraction.
Okay, do you normalize for the amount of IMEC beads then in that case, or do you just
No, in that case, because since it's very low amounts, we prefer to normalize for what we think is the highest in all the fractions, and we use the same, just to be on the safe side.
Okay, so I've got two hands here. Let's see if I can more allow us to unmute. So Vitautas and Ruth.
Can you hear me?
Here we go.
Yes,
you can hear
Great.
you. Hi.
Thank you so much, Anna, for a great talk, and Stoyan for organizing this event. Sorry if I missed. I would like to ask, how much starting material do you recommend to use, and what would be lower and upper limit for this? So that would be one question. Shall I wait for the answer? And
well, I can answer you now. Great. It's such an easy one. We use 1 P15 as input material, and I don't think why not. You cannot scale it down. So for proteome, you should easily scale it down. For phospho-proteome, maybe if you scale it down, you start to lose some sensitivity. For tissue, for the data I saw, we use a full kidney, but for the liver, we only use like 1/3 of the liver. And scaling it up, that we saw that if you start to pull samples and the fractionation doesn't work as well, because you need really to have the cells moving perfectly. So probably if you want to scale it up, you also need to scale up the volumes you use for fractionation. But as the protocol was published in bioRxiv, it's 1p15, 70% confluence.
Yes, so that would be one plate for the phospho-proteome and one for proteome.
No, the same.
The same. Another question would be how reproducible is the fractionation method? Is it set up on the robot at the moment, or is it done by hands?
So the fractionation is done by hand because it's based mainly you add the different buffers, you incubate, and then you pellet, and then you recover the supernatant. So the proteins released in each step will be soluble, and you will get them, and then you continue with the pellet. That so far we need to do it manually. But
so based on your experience, do you feel like you need quite some training to achieve?
No, you just need to know how to pipe it.
Sounds easy enough. So maybe the last question.
It's a complex. it's not like doing a dilution. It's a protocol with several steps. But from the technical perspective, I don't think it will take you more than 1 day to get used to it.
Yes, and the last question: have you considered using TMT labeling for such an experiment? And how would you set up then?
So that's one thing we don't want to do, because when you do TMT, the only way you have to get deep depth on the analysis is to do fractionation. And what we want to do is to keep the acquisition time short so we can include a lot of conditions. Because if we label with TMT the different fractions, for instance, with a 6-plex, we will have a TMT 6-plex for each condition, like time point. But if we just run single shot analysis with that, the depth of our coverage will be very poor than if we do single shot DIA analysis. So that's why we go for DIA single shot and we don't go for TMT labeling.
I understand. Thank you so much for answers.
Yeah.
And so we still have Ruth's hand, but you're gonna have to activate your mic, Ruth. Otherwise, if you can pose the question, we can get to it in the chat or in the Q&A section. Okay, no. So, okay, I don't think there's any more questions. So Anna, thanks again. It was really awesome to have you for this episode too, and we appreciate you coming to chat to you about the work you're doing.
Thank you for inviting me.
No, it's a pleasure. And thanks everyone for attending, and look out for the subsequent episode, and the recording will make this available in the next couple of days. And have a nice day further. Cheers.
Yep.
Bye.